Swayam Prabha. Digital India. Educated India.
Welcome back to the second lecture of week 3. So, as I told you in the previous lecture while starting the week 3 that next 15 classes will be dealing with muscle, nerves, and somewhere in between we will talk about a small tail piece of conjunction of skeleton and the muscle in the form of appendicular and axial skeleton and appendicular and axial muscle and the articulations. Okay. So, if you recall back, so where we stopped in the last class. So, I gave you 3 examples: beating of your heart, you playing basketball or whatever games you like, where your muscle is getting a lot of stretch, and the third thing, you are eating food. And I told you that these 3 are orchestrated by muscle. The muscle lining your alimentary canal or digestive tract, which is termed as the smooth muscle. When the bolus of food moves along this alimentary canal or the digestive tract, you don't even feel that there is something moving along your body. Okay? It is so so subtle and so smooth out there. Yet when you take your hand and touch out here, you feel there is a lubbed up sound, and if you can't feel like that, you just put a stethoscope, you will be able to feel it. So there is a big chunk of organ which is beating, and there are no bones in it. It is suspended out there in your thoracic cavity, which is called the heart, and these muscles which are having a lot of stretch. And I told you the fundamental difference between them, if you look at from a very holistic point of view, is the force generated by them and the force which is directly linked to the displacement they can do. You can stretch most of your skeletal muscle. So, one underlying theme what we came is, it's not all muscles cover the bones, point 1. Second, these 3 different muscles have different force dynamics. And we concluded the class saying that in order to understand what are those dynamics, one has to understand the development of the muscle in the first thing. Once you understand, just like the straight corollary is that when we had to talk about the bone, we had to understand how it develops from the cartilage, because initially it was the cartilage. Okay. So, now let us move on to understand the development of the muscle. So, today we are into week 3 and this one is lecture 2, right, lecture 2. So, W3/L2, this is where we are. So, now talking about the development of the muscle. So, I told you in the previous class that the classic example what we will be taking will be of the skeletal muscle. Okay. And later, as we will be talking about the cardiac muscle, we will talk about the features of the cardiac muscle. And while we will be talking about the digestive system, we will talk about the feature of the smooth muscle. So, muscular development, just like the way cartilage develops pretty early in the mother's womb, muscle development also happens pretty early. So, there are, when the embryo develops, there are certain class of cells which are destined to become muscle. Okay. So, these cells migrate to their specific places. It can be on top of the bones, since we will be talking about not bones, the cartilage precisely, because it is the cartilage from where the bones are formed. and yet some which forms the beating organ, the heart, and the ones which form the alimentary canal. What happened after that? So those cells, so say for example, I say, so this is a cartoon of a human being, right? So say for example, so when we are in our mother's womb, like say for example, I blow it up, something has to form a, one second, something has to form a muscle here. So what happens here? So those, say for example, in this embryo, which is a mass of cell like this, okay, among these mass of cell, say for example, some of them, a lot like this is destined to become muscle, or let's say they are destined to become skeletal muscle. Since we are taking the example of skeletal muscle, we'll talk more about the skeletal muscle at this stage, okay. Now these muscles, they're destined, okay, they haven't formed yet, they're destined to form skeletal muscle, okay. So these cells, so there is a set of cells which migrate, I'm just putting those cells in red, the one which are shading them red for your understanding, okay. These cells migrate to different parts where the skeletal muscle has to form. This is the first level of differentiation which happens. They are moving to different zones. So just for the simplicity's sake, I'm just keeping, okay, something like this. These are the 4 different locations. I'm just putting there so many locations. All the bones which are there, they must have moved. Okay. So they have migrated. And the first level is migration of the cells which are destined to become skeletal muscle. Fine. So these are different sites. So just I'm putting the blue bordering as S1, S2, S3 and Sn. Okay. These are different sites. So now what we will try to figure out, what is happening that at specific sites. At specific sites, these individual cells initially, these individual cells reaches the site, and at that particular spot they start to divide. This is the first thing which happens. They form a kind of a lump of cells like this. Okay? They're dividing like this at that specific site. Right? See, they are now in a dividing phase. And I'll try to get an animation which I once make long time back for you. So first and foremost thing is they undergo a massive division. Step 1. So in order to remember in your brain map, put the first sign, the division. After division, something very interesting happened. So this whole thing has divided, and then they started to form kind of rows like this, something like this. Just follow my pen here, you will realize what they do next. Next, there is something called alignment. So these cells started to align like this. It's a very interesting thing. I mean, if you see it under a tissue culture dish, it just looks amazing the way they align it. I had the privilege and great fortune and blessings to do these kind of experiments. So I've seen them in a real life, and it just in a dish they look phenomenal, like this. They started to align. Okay. So while I am drawing this up, it should strike, the first thing which should strike your mind is, what are those molecules which help them to form this kind of alignment? It's almost like a rope, as if they're aligning like a rope. Those of you who are from villages, you must have seen when this jute or different hemp or something, they form a kind of fiber like this. They pull the fiber out of it, almost like you're pulling fibers. These cells align. So let's go back. What all we did? In the embryo, there are cells which are destined to become muscle. Step 1. And those cells which are destined to become muscle migrate to their specific locations wherever they have to form a skeleton. In this case, we are talking about skeletal muscle. We'll talk later about the cardiac and the smooth muscle. So at this point, just concentrate on the skeletal muscle. Those cells migrate to that specific region. Say, for example, something has to form these muscles of my body, right? So they will migrate here. They will, scattered around the bone or the cartilage which is present at that stage, whatever stage of development that it is, they're sitting on. Then these small lumps which will be formed, they started to divide, as I showed you. So if you see it here, they are dividing. That's why I kind of put that signature for you. They are dividing. After dividing, they are moving for the alignment. They are aligning in a line. Aligning in line. This is a beautiful alignment which is happening, Now, what I was trying to tell you, it must strike you, what are those molecular players? Who tells them that you have to align like this? You won't align like this, you'll align like this. These are some of the biggest challenge of modern biology. Who tells them all these things? How they know they have to align like this? Did it strike you? It should strike your brain that, there has to be certain molecular players which are present here. They have to be present here, who are telling them, this is the guidance cue. These are called the guidance cues or guidance molecules. Now you realize that why those first week was so important when I'll tell you. I told you that all these organizational cues are very important. Who tells them? There has to be some form of a guidance, otherwise they cannot align like this. Okay? So this is the first level of self-assembly which happens. They align. The next thing what happens is even much more interesting. They have aligned. Now, these individual cells which are present there. Now you see this. Now let me go to the next page to tell you exactly what happens. So I drew it up to this, right? These are the individual cells which are present out here. Okay? With their individual nucleus, and they are aligning like this. Now, at this stage, there are two things which happens. So I showed you. So they have aligned like in a longitudinal manner. Some of these cells even further divide while in an aligned state. Further division happening. It's almost form a three-dimensional rope kind of thing. And then it's division stops. There is no more division. Division ceases. There is no further division now. Now what happens is there are two cells which are touching each other. You see this? So look at this zone. I am drawing in pink. You see this? There are contact points, contact points, contact points, contact points, right? I am circling them. These contact points are lost. What does that mean? That means instead of having individual cells like this attaching together, this whole thing becomes one common. Almost if you keep on adding them small cells like this, and the contact point are lost, what it looks like, it will look like a complete tube. So think of it. Now let me go to the next level. Like, okay, now these contact points are now getting merged. I am showing it even now orange color. These contact points are lost. So what you essentially will be landing with is something like this initially. As if it is like a tube-like structure. And with the individual nuclei, as if it forms a multi-nucleated structure, a multi-nucleated tubes. And there is no, you won't be able to figure out which cell is which one. So what you get is a multi-nucleated tube. And there are several such tubes which are formed, and it's a three-dimensional tubes, as I told you. They are almost in a 3-dimension form like this. Okay? There are several such multinucleated tubes. So now out here you have such long tubes. So that's why I give you the analogy of those of you who are from village or goes to village or goes to rural places where they pull these strings, from hemp, from jute, from any sources, and in India there are several places, or even those where they put the silk threads. You'll see how they are formed. They have individual threads and then they pull them together, they turn them, coil them, supercoil them, and likewise and so on and so forth. It's almost like you have thin tubes like this with lot of nucleus in them, with multinucleus. And nuclei are arranged, nuclei are arranged like this. Okay? Next thing is another interesting thing which happens. Once these tubes are formed, these tubes come close to each other and form something called exactly what happens in the formation of the thread. They form a supercoiling. So in other words, these different, so now I am just showing it in one line, the individual tubes I am showing in one line, okay. These, they come like this, they form a supercoiled structure like this. And will come, there are specific names for it. So these are lot of tubes which are coming close to each other and forming a supercoiled assembly. Right? Doesn't it look very interesting? So this is how, in case of skeletal muscle, it happens. Now, we haven't talked about anything about the molecular players, but what we figured out is that at this stage, also, the first I told you in the previous slide, if we go back to the previous slide, there are guidance molecules from here. The guidance molecule which guides these individual progenitor cells to move to their specific sites, and out here they get a signal to divide. So it means there are dividing molecules which are present there, okay, dividing molecules, and these dividing molecules further in conjunction with the guidance molecules tell them to align in a specific line. There are further division or may not be further division, but the next thing what we know is that these division molecules, dividing molecules stop. There are no more dividing molecules are allowed, and what you get is there are molecules which pitches in here, which tells, so this is a new set of molecules, that these are another set of differentiation molecules which tells these muscles or tells these muscle cells to lose their identity and form what we call as a tube-like structure, what we termed as the multinucleated tube. So at different point we see different differentiation molecules or different molecular players which are doing it. Next what we know, there are another series of signaling which is coming, molecular signals, which are telling them to form a supramolecular coiling. Okay? So you see that every point there are unique signal molecules, guidance cues, dividing signals, non-dividing signals, merging signals, coiling signals which are involved in it. And that's what the whole molecular physiology is all about, deciphering, discovering these things, because any mistake in any of these things could lead to difficult diseases like muscular dystrophy and so many other things. Okay. So now, what are the scientific terms for all these things? Still, and by the way, I still haven't touched upon how the force is generated. Okay, I will come slowly into it. First of all, you have to get a mind map of what is happening. Now let me introduce the scientific terms for each one of these. Now let me go back to the previous slide. These cells which are destined to become muscle are called, anything to do with muscle is designated by this word called myo, M-Y-O, myo. Okay? And anything which is dealt with cell is called site. So these cells are called myocyte, muscle cells. Okay? Clear to you guys? Next, these myocytes align together, divide, these myocytes divide and then align together. There is alignment of the myocytes, as you could see in the previous slide out here, alignment of the myocytes followed by formation of a multinucleated tube. And this multinucleated tube is called myo, coming from muscle, tube. Right? Now these different myotubes align with each other at different level. Some of them merge like this, there are merging like this, as well as there are merging like this. So end to end they are adding together or merging like this, forming a supercoiled assembly. This formation is called myofiber. So see, we started with the progenitor cells which went to their specific location in the cartilage during development, followed by the generation of series of myocytes, CYTE sites, the cells which are destined to become muscle. These myocytes align with each other to form a multinucleated structure called myotubes. These myotubes with respect to myocytes, have lost the individual identity of the cells because they all have coalesced together and form a tube-like structure. These myotubes align with each other both at the proximal and rear end, like this, tube, to multiple tubes attached together, as well as from sidewise. So they have a sideways attachment, they have an attachment like this, right? One on top of each other, as well as like this. And then they form a supercoil assembly like this, okay? And those supercoil assemblies are called muscle myofibers. And these multiple myofibers, this integration or the summation of these multiple myofiber form what you call as muscle tissue. Now, going back to the first slide when I told you that if I now look at this one. So, we started with the cellular players, we talked about the formation of the tissue, and eventually it formed the organ. You see, at every level there will be an organizational setup. But mind it, what I have as of now I haven't touched, because I'm going slowly on it, because is who are the molecular players, right? Because it is the molecular player who will determine. So I have given you some hint about the molecular players out here, but those molecular players who are involved in as a guidance molecule, as a dividing molecules, okay, as these differentiation molecules, but still I haven't told you who are those molecules, which are those molecules which are involved in the process of generating different kind of force. So, what we will do in the next class, in this structure, we'll again revisit this whole development process, right? We'll revisit this development process and we will talk about those molecules which are involved in generating the forces. And based on that, one can classify the muscle at different points. Even the skeletal muscles are further classified into two forms of muscle, which will come later, but just for your understanding's sake, because whenever you consult a book or something, you will come across these two words, okay? Extrafusal muscle and intrafusal muscle. These are both are classifications of skeletal muscle. We'll come into this, what are extrafusal muscles and what are intrafusal muscle. But at this stage, and do not get confused, please do not get confused. The whole idea of this course is to kind of, make things simple for you, so that if you pick up any book, you should be able to understand that what you are really looking at. And most important for me is you understand this organizational setup. This organizational setup is very important. How much soever simple it looks like, it is here. If you understand this whole ball game, you'll be able to develop your own stories. You should be able to formulate your own research problem. Okay? So, what we will do next is, what are the individual molecular players which are present in myocytes? The first one is the myocyte, right? Which over a period of time get transformed in order to distinguish a myotube form for skeletal muscle, a myotube form for cardiac muscle, a myotube form for smooth muscle, or even within the skeletal muscle, a myotube form for extrafusal fiber, a myotube form for intrafusal fiber or intrafusal muscle or extrafusal muscle. So, today, if I had to summarize what we did, we talked about the cellular differentiation, how the cells join together to form a tissue-like structure or a tissue called muscle tissue, right? In next class, what we will do, we will talk about the molecules which are present in these muscles which led to the formation of the tissue and thereby giving it that feature what we talked about in the very previous class is force generation and correlating it with the molecules of muscle cell, actually let's call it muscle tissue. Okay. And all these things will all happen at the level of myotube / myofiber. Okay. So let's again take the clock back. Myocyte, this is one technical terminology you should remember. Myocyte lead to the formation of myotubes. Remember that? Different myocytes attached together. And these myotubes join, align together, sometime like this, on top of each other, as well as from sidewise, form myofibers. And multiple myofibers from muscle. Okay? Keep this in mind, and this framework is good enough for the rest of your life to develop the story about muscle. Okay? So next class, we will talk about the molecular players who are involved in this game. Thank you.